Hello again, class. Now we are moving on to the large intestine. So we started at the mouth, went to the pharynx, the esophagus, the stomach, where we started with the protein digestion. Uh, at the mouth, we had the carbohydrate digestion and mechanical digestion. Stomach, we have more mechanical protein digestion. Then we go into the small intestines, duodenum, jejunum, ileum, and now we go into the large intestines. All right, so large intestines, otherwise known as large bowel, that's where the term bowel movement comes from. It comes from your large, that's another name for your large intestines, large bowel. So when we use that term bowel movement, we're basically talking about emptying some of the feces which are in our large intestines. All right, so the large intestines runs along the border of the abdominal cavity. As you'll see in the diagram, sort of surrounds your small intestines and other abdominal organs. About 5 feet long. Notice that it is significantly shorter than the small intestines, but has a much larger diameter than the small intestines. All right, so that's something to make a note of. Significantly shorter, only 5 feet long, but so only about a quarter of the length of your small intestines and lengthwise, but diameter has much more space. Okay. The small intestines feeds into the large intestines, and it's the passageway for feces to exit the body. So we have a lot of absorption of the nutrients from the food that you eat in the small intestines, and then once we get to the large intestines, it's going to be more about preparing what's left over to be removed from the body. Clearly, everything that we eat, we do not absorb. And then also our body creates solid waste. Remember, waste is also created by the cells. So there is large intestines is also active in absorbing water and electrolytes. We're going to talk about that. And there's the task of the large intestines to have some secretion and propulsion, got to move things along, and ultimately defecation, which is bowel movement, removing it from your body. There are also numerous bacteria that live in your large intestines. So they're technically organisms, bacteria, single-celled organisms that live in your large intestines, which are also very interesting. And we need them. They're there for a purpose. All right, so once we get past the stomach, the duodenum, the jejunum, the ileum, we get to the first part of the large intestines, which is the cecum. Right, cecum, first portion of large intestines, is known as a blind pouch located in the right lower quadrant of your abdominals. Right lower quadrant, can't really see this on the picture. Right lower quadrant. Features a smaller blind-ended pouch extended from its posterior inferior end, generally shortened to simply appendix. The appendix houses multiple lymphatic nodules, plays a role in the immune system. So that's going to be, we talked about the appendix a little bit, but and the role that it does have a role in our body. We can remove it, like we talked about in the last section. Not the cecum. We can remove the appendix, not the cecum, and it plays a role in immunity, but it doesn't play a role that if we remove it, we won't. It's not life threatening. It's life threatening if it becomes infected, but not removing it. You'll see all this in the diagram that's coming up. The colon, the next and longest portion of large intestine, divided into 4 portions. And this is what the colon is, what we think about when we're thinking about the large intestines. ascending, going up, travels superiorly on the right side. And then it reaches the liver, makes a sharp left-hand turn. It sort of looks like almost like like you're trying to draw a square. Sharp left-hand turn at the hepatic flexure. And then at a hepatic flexure, ascending colon becomes intraperitoneal, transverse colon. Then at the spleen, it takes another sharp turn, becomes at the junction called the splenic flexure. And then we give rise to the retroperitoneal descending colon and passes along the left side of the abdominal cavity. In the left lower quadrant becomes the S-shaped sigmoid colon and intraperitoneal and passes toward the sacrum. Remember the sacrum at the base of your spine. All right, so here we have the cecum, as you see the cecum here. That's where we're connected to the small intestines, to the small intestines. The ileum, the smallest, not the smallest, the ileum, the last section of the small intestines, is connected to the large intestines at the cecum. You see the appendix right off of the edge of, at the edge here, off the edge of the cecum. That's when we say when you have appendicitis and we need to remove your appendix, that's what we're removing. But it does have some ability to help remove some, to act in our immune, as far as our immune system is concerned. Then we get into once we get past the cecum, then we get into the colon, where we come up, ascending, transverse, coming across, and then coming back down, descending colon. And then we have this curve here, sigmoid colon, right? And then eventually we have the rectum, which is going to lead to the anus, right? So after the sigmoid colon, let me just move my picture. After the sigmoid colon, large intestines continues as rectum and runs anterior to, anterior means in the front of, anterior to the sacrum, and is retroperitoneal, talking about in regards to the peritoneum. The walls of the rectum feature horizontal folds, rectal valves, right? So there's valves in the rectum, which are important, which allow you to be able to release gas. Flatulation is what that's called. You see the word flatus here, but flatulation is the ability to release gas without risking simultaneously passage of feces. So you can release gas but not feces. That's an important factor in the rectum. And the rectum is basically the final passageway of the large intestines, and that ends in what we call the anal canal, which is considered the last portion of the large intestines. That's basically when things are coming out of your body. Feces are coming out of your body. The anal canal passes through the levator ani muscle in the floor of the pelvic cavity. Notice something that's important to notice here is that this is where we go from the smooth muscle back to some of the back to skeletal muscle, which is very important, obviously. Unlike some animals who are free to make bowel movements whenever they feel like it, out in the wild, basically. If you see horses walking down the streets of Philadelphia, the horse and carriages, they just kind of go just whenever it gets to that point where they need to go. There's no sense of control in that sense. But as human beings, we have the ability we have this voluntary external anal sphincter, right? So After your feces passes through your anal sinuses, you have this involuntary internal anal sphincter, which is that's involuntary. That means you have the muscularis externa. That means smooth muscle control supplied by your parasympathetic motor neurons. But then we also have this voluntary external anal sphincter. That means it's composed of skeletal muscle. That means we can control it consciously with our minds. This sphincter is voluntary, supplied by somatic motor neurons controlled by the cerebral cortex, our brain. All right. So there's involuntary and voluntary control at this point. So at the very beginning, we're chewing food using the tongue, we're swallowing, voluntary, and then slowly switches. Basically, once you get into this esophagus, gets to involuntary. Esophagus, stomach, involuntary, the propulsion, the peristalsis, the motility, and the intestines, all involuntary. Squeezing, churning, all involuntary. Large intestines, involuntary. And then we get down to the anus, and that's where it becomes voluntary again. So voluntary in the beginning, what I choose to put in, how do I choose to chew my food and swallow my food, voluntary. And then at the end is voluntary, but then everything else in between is involuntary. Very important point to notice. And this is what the structure of the rectum looks like. Rectal valve, anal canal, see the levatory and eye muscles, internal sphincter that has that smooth muscle controlled, external anal sphincter. These external sphincters are controlled by skeletal muscle, voluntary control. And then anus, and that's the exit from your body. Right? What is the histology? We have to look at the cell structure of the large intestines. We have mucosa of large intestines, which lacks villi, and the cells lack microvilli. So it's not adapted for absorption. That is not the primary function. So that is a major difference between the large intestines and the small intestines. Small intestines has the villi and the microvilli, the enterocytes that really focus on absorbing nutrients from the food that we've eaten. Whereas we don't have that high surface area in the large intestines because we do not want, at this point, we're not doing a whole lot of absorbing. The mucosa is rich with goblet cells that secrete protective and lubricating mucus. We've seen that already before in different parts of the respiratory as well as the digestive system. The muscularis externa of the large intestines is unique. It's not longitudinal layer, is not continuous throughout most of its length. It's gathered into bands, 3 bands of ribbons or muscle. They're constant tension bunches, colon in the pockets. So you have these 3 bands. That's how they're gathered. The musculature is a little bit different in the large intestines. The serosa, the visceral peritoneum, the outer surface of the large intestines contains fat-filled pouches. So it's a little bit different structure than the small intestines. It's not necessarily as designed for the same type of motility as the stomach or the small intestines are. Right, but now we're going to get to the bacteria, which we mentioned earlier. Large intestines host all these bacteria, which it seems kind of. I remember when I first learned about this a long time ago, I was like, "Wow, that's interesting." So we have bacteria that live in our bodies that are supposed to be there. There are about 10 times more bacteria in large intestines than cells in human body. Bacteria make up as much as 60% of dry mass of feces. This is why, well, this is one of the reasons, there's multiple reasons, but this is one of the reasons feces can be so dangerous, is because you don't want these bacteria just anywhere. And so if you word, not to, I know this is disgusting, but not that anybody would ever eat feces, but little kids sometimes, and I have little children, they will put things in their mouth that should not go in their mouths because they don't know better. And so this is why it can actually be dangerous to have feces in your mouth, because you've put bacteria that is not supposed to be anywhere but your large intestines. You've introduced it to different parts of your body. So these bacteria live in your large intestines and make up about 60% of the dry mass of feces. These bacteria, which are called normal flora or gut flora, 500 different bacteria species, they exist as in a symbiotic relationship. If you've ever seen the movie Venom, maybe you've seen that movie. If anybody out there is a fan of superhero, I guess, movies, that the character, which I forget his name, there's this like alien creature, and they live together as one. Like the alien can like work through him. It's like and they called the alien a symbiote. But so symbiotic simply means that two separate organisms living together, benefiting each other. So the bacteria get something that benefits the bacteria in this case, and we also benefit from the bacteria. All right. And what do they do? Well, I have it right here on the chart for you. Humans provide bacteria with the environment they need to survive. They perform a number of functions. They help produce vitamins, such as vitamin K. They metabolize undigested materials, so some carbohydrates that small intestines is unable to digest. They can convert them into fatty acids. They deter the growth of harmful bacteria. So these good bacteria stop the growth of harmful bacteria. And then they can also stimulate the immune system, right? So during infancy, the normal flora induce immune tolerance to their own antigens. At the same time, they stimulate development of MALT, mucosa-associated lymphoid tissue, and production of antibodies to pathogens, right? So they're there for a purpose, and they live inside of us, and that's how we exist. All right, now let's talk about motility. Large intestines has two functional segments: proximal large intestine, which is the ascending and transverse colon. So ascending, transverse, and then the distal large intestine, which is the descending sigmoid, where it curves, and then down to the rectum and anal canal where it comes out. Proximal large intestine is primary site of water and electrolyte absorption and bacterial activity. All right, so your gut flora, and also have water and electrolyte absorption. And there's two different types of motility involved, right? So we have one. Remember, once again, everything has to move in the right direction, continuing from the mouth, esophagus, stomach, duodenum, jejunum, ileum, cecum, to when we get to the colon and rectum and the anus. All right, so know that whole pathway, and that once you introduce food into that pathway, it has to keep going in the right direction. At no point do we really want it to go backwards. Not really. All right. There is some churning back and forth in the stomach and things like that, but for the most part, we want things to move in one direction. All right. So in the large intestines, we have segmentation, which is a churning. So remember the three layers of circular smooth muscles, swirls material around. This aids in the water and electrolyte absorption. and it's controlled by your ENS. We have another type. We have mass movement, mass peristalsis, which is like a propulsive motion. So propulsion, moving things along, all right? During mass movement, multiple hostile undergo peristalsis, prepares their content toward the distal large intestine. So it's just pushing this soon-to-be fecal matter right along. All right, this happens 3 to 4 times per day, triggered by your food consumption. Initiates reflexes controlled by your nervous system, your ENS. The distal large intestine performs a small amount of absorption, primarily of water, but its main role is to store fecal matter, to store feces until we are prepared to have a bowel movement. So it's stored primarily in your distal large intestines. Remember, that's distal is the descending, the sigmoid colon, the rectum. It's where it's stored, right? The distal large intestine is much less motile than proximal. So when mass movements force fecal material into normally empty rectum, it initiates this parasympathetic mediated defecation reflex. So when your rectum starts to is stretched, there's fecal matter in here. So normally this area is empty, but now your body's pushing fecal matter into this area. We get this defecation reflex. This stretch receptors transmit sensation to rectal distension of the spinal cord, causes smooth muscles in the sigmoid colon and rectum to contract. Right, that's at the sigmoid colon, the last curve of your large intestines and your rectum, leading to your anus to contract. Internal anal sphincter relaxes. You feel this. You start to feel the sensation of feces, and impulses from the cerebral cortex trigger relaxation of external anal sphincter and contraction of levator ani muscles. Right? And so that's the process of wanting to remove feces from your body. So it's basically based on a stretch reflex. As the distal large intestines, as the rectum starts to stretch due to fecal matter, it sends a signal and basically lets your body know you got to go to the bathroom. All right, so this is that process. Stretch receptors transmit sensation. Parasympathetic neurons cause smooth muscle in the sigmoid colon and rectum to contract. Impulses from the cerebral cortex trigger relaxation of external anal sphincter. That's the part that's controlled by your external anal sphincter, is smooth muscle, that's conscious control. Contraction of the levator ani muscles, allowing feces to pass out of the body. All right, so anything that changes motility of large intestines affects amount of water present in feces, right? And you've all, I'm sure, probably had this experience when motility increases. So when something moves faster through your large intestines than it's supposed to. Large intestine does not have time to absorb water from the fecal material. Remember, when we come from the stomach, we go from the bolus to the chyme. It's basically a fluid by the time it enters your small intestines. Remember that. So, but when we go to the bathroom and we have a bowel movement, that feces is a solid, maybe a soft solid, but it is a solid, right? So how do we go from eating, which is a solid, creating a bolus, and then going to a chyme, which is a fluid, and then we go back to a solid? So what happens is a lot of the changes is because of water, the amount of water introduced. And so when we're in the large intestines, that water needs to be absorbed, therefore, and which helps turn it back to a solid. Now, if something goes through your large intestines faster than it's supposed to, the water doesn't get reabsorbed by your ascending colon, right? And so what happens is when you go to the bathroom, the normally solid fecal matter has more water in it, has more fluid in it, and it is what we call diarrhea. All right, watery feces. Hopefully that makes sense. And this can be due to irritation of the colon, bacterial viral infections. Also, certain drugs can stimulate your body to stimulate the parasympathetic nervous system and cause fecal matter to move through your intestines faster than it's supposed to. The reverse can also happen. So something feces spends too much time in your large intestines. Therefore, you have more water absorbed from that feces than normal, and it becomes hard. So it becomes, notice we like it to be kind of like, soft, a little solid, but kind of soft. But if too much fluid gets absorbed from that fecal matter, it moves too slowly through your large intestines, then we get what we call constipation. It becomes hard and it becomes difficult for it to pass through your descending colon, sigmoid colon, and your rectum and your anus, and it can actually become painful. And this is also can happen based on drug use, specifically opioids, and opioid narcotics can cause severe constipation, can cause people not to go to the bathroom for days or a week or longer, and that can be very uncomfortable for the person who is taking these drugs. All right, so this is just a diagram just showing you, and I think I've said it over and over and over, but this is just kind of showing you the whole structure of your digestive system. Remember that there are other organs involved in the digestive system that are not directly part of the alimentary canal. Remember, we have a couple of other organs, which we have listed here: the pancreas, liver, and the gallbladder, which play an important role in digestion. But food per se, they're not technically on the pathway. They're connected to it, but they're not in the lumen, the canal that the food directly passes through. All right, so we have pancreas, liver, and gallbladder, accessory organs. Other accessory organs that we've talked about are the salivary glands. Remember those up in the oral region that release the initial enzymes that help with the digestion of polysaccharides, breaking them down. The pancreas, liver, and gallbladder are exocrine glands. They secrete product through duct to outside the body. Now, this sounds a little bit confusing because if you think about it, if I think of the alimentary canal as like if you look at your hand and make a circle, put a hole in the circle, put a circle. Now, if something technically passes through this circle, it's not really going into your hand. Now, if something went into the skin this way and that would be into my hand. But if it's just passing through a circle, it's not really going into my hand. that is like think of that as like the alimentary canal. If I were to swallow like a tiny marble, it would pass through down my throat, my pharynx, my esophagus, stomach, and small intestines, large intestines, and it would come right out of my anus. That small little, if it was a like a stone, small little marble, would come out pretty much the same, untouched. It wouldn't be comfortable necessarily, especially depending on the size, but it wouldn't be absorbed by my body. So technically, until something gets absorbed, we think of the alimentary canal like this, like it could pass basically as an opening through your body. Until it gets absorbed, that's meaning, it's not really in your body until it gets absorbed, right? So when we say the pancreas, liver, and the gallbladder are exocrine glands, they secrete product through ducts to outside of the body, even though they are secreting to inside of the alimentary canal is technically considered outside of the body. And that can be a little bit confusing. It's because that canal, as it says here in that first point, is basically open to the external environment on each end. Right? All right, so let's go into we're going to talk about each one of these, the pancreas, the liver, and the gallbladder. First up is the pancreas, and the liver and the gallbladder are sort of connected to each other, so they're very much related. All right, so the pancreas is a gland with endocrine inside and into the body, and exocrine outside functions. Endocrine, hormones, insulin, helps with our sugar, glucagon, helps with our glucose. It can affect most cells in the body. We talk about those when we talk about metabolism. Exocrine, secretions, enzymes secreted primarily by a cluster of the acinar cells and released into ducts of pancreas that empty into the alimentary canal. And we've talked about kind of where these enter already, near the duodenum. All right, so pancreas, upper left upper quadrant of the abdomen. And it has a couple different sections. The head is, three sections. It has a head section, which is a wide section. That's in contact with the duodenum. Remember the duodenum, the first part of the small intestines. Then we have the body, separates the head from the thinner tail, and tapers off towards the spleen. We have the main pancreatic duct, travels down the middle of the pancreas, receives the secretions from the acinar cells. And then near the duodenum, the main pancreatic duct merges with the duct from the liver, which we're going to talk about right after this, and gallbladder, and empties into the duodenum. So the liver, the gallbladder, the pancreas all basically enter their exocrine secretions into the duodenum. Each acinus, which is a collection of acinar cells, surrounds small duct into which its cells secrete their products. And these small ducts merge and drain to the main pancreatic duct. And although some secrete into a smaller accessory pancreatic duct. And so that's the basic structure of the pancreas. You see the liver here, and you see the gallbladder, and then you also see the pancreas, all near. You see the stomach right there, all near the stomach and the duodenum, the first section of the small intestines. The duct from the liver and the gallbladder and the pancreas all merge together, and we get the exocrine glands releasing their hormones into the duodenum. All right, right into the duodenum. I mean the enzymes into the duodenum. All right. All right. So what we want to know exactly what those are. So we have what we call pancreatic juice. It doesn't sound very like a very creative name, but that is the collection of secretions of pancreatic acinar and duct cells made up of water, multiple digestive enzymes. This helps with digestion, and a couple other proteins. Duct cells also secrete bicarbonate ions. So bicarbonate, remember that that bicarbonate formula helps balance pH. So these bicarbonate ions make pancreatic juice alkaline. Remember that the chyme that is coming out of the stomach that's coming into the duodenum is very acidic. This pancreatic juice is alkaline. The alkaline juice helps neutralize the acidic chyme that enters the duodenum. All right, and so then these digestive enzymes also secreted by the pancreas, the acinar cells, are also crucial in chemical digestion. They catalyze reactions that digest carbohydrates. We've already talked about carbohydrates and the salivary amylase in the mouth. Proteins, we've talked about digestion of proteins a little bit already with the pepsinogen in the stomach, but then also helps catalyze reactions to further digest lipids and nucleic acids, which we haven't really talked about up until this point. Breaking them all down further. Pancreatic secretion occurs at a basal rate between meals. So even when you're not eating, you have some pancreatic juice secretion. But during eating, it rises due to parasympathetic and hormonal stimulation. So, which makes sense. As I eat, I need more pancreatic juice to do more, to help digest the carbohydrates, proteins, lipids, and nucleic acids that I'm eating. You're going to see this CCK. We're going to mention this a couple times. Cholecystokinin, which is produced by the duodenal enteroendocrine cells, okay, in response to presence of lipids and partially digested proteins in the duodenum. So the first part of the small intestine senses that there are partially digested proteins, which we've talked about as far as we mentioned those and setting off triggers in the stomach as well as in the duodenum. This acts on the acinar cells to trigger secretion of digestive enzymes and other proteins. Right. Then we also have secretin released by the duodenal cells in response to acid and lipids in the duodenum. Remember that acid means that something's coming from the stomach to the duodenum. That means food's involved. And lipids also, there's a lot of lipids as fat that's going to be in the food that you've eaten, can trigger these duct cells to secrete bicarbonate ions. All right, so CCK and the secretin help these help to elevate the pancreatic secretions. All right, so CCK secreted, secrete secretin, CCK, those are secreted, those stimulate your pancreas, they make the pancreatic juice. All right, now we move on to the liver and gallbladder. There's a lot of information on the liver. It's a very it's an organ that obviously very useful, that we are very much in need of. As someone who has, I have a family member who struggles with alcoholism, and in that struggle has had problems with his liver. And you don't want problems with your liver. Now, it is true the liver is one of the few organs that can regenerate, but the liver does a number of things, and so you ultimately want a healthy liver, and you're going to see exactly why. Okay, so the liver is, as you see here, pyramid shaped, located in the right upper quadrant, just inferior to the diaphragm. Right, we saw it in the other diagram, near the stomach. Remember, the esophagus passes through the diaphragm. You have the stomach, the pancreas, and the liver are kind of all right there. The gallbladder is just a small sac on the posterior side, sort of the bottom side of the liver. And they have a close anatomical and functional relationship. So they work together. All right, so the anatomy of the liver is a little bit complex. It's covered by a thin connective tissue capsule and visceral peritoneum. We talked about the peritoneum, the visceral peritoneum, in relation to the mesenteries and the peritoneum. The liver is composed of 4 lobes, large right and left lobe on right and left sides of the liver, respectively, separated by a fold of the visceral peritoneum. You have this round ligament on liver's inferior surface, remnant of the umbilical vein. Then you have the two smaller lobes, the caudate lobe and the quadrate lobe on posterior side of the liver's right lobe. All right, so you have those 4 lobes. That's just giving you sort of the shape, structure of the liver. And then we have this porta hepatis, or hepatis, whatever you want to say it, an indentation on the liver's posterior side where numerous blood vessels enter and exit the liver. All right, this is going to be very important. Includes the hepatic artery, hepatic portal vein, hepatic and anhepatic veins. Nerves, lymphatic vessels, common hepatic duct. All right, these are all, remember, the liver needs its own blood supply, but it's also we're gonna see this play a huge role in digestion. Okay, this is the structure of the liver: right lobe, left lobe, right and left sides. You see the gallbladder, the round ligament from the umbilical cord. Flip it over, posterior. Okay. Quadrate lobe, gallbladder. Caudate lobe, right? All right. All right, liver lobule, right? Basic unit of liver and here are the histological features, all right? Separated from one another by septa that branch and form a connective tissue capsule of liver, composed of flattened plates of cells. This is the key. Hepatocytes arranged in shape of a hexagon, and stacked on top of one another with small central vein in each lobule center. Right? All right, hexagon is the shape. You have these flattened plates of cells stacked on top of one another, a small central vein in the center. Right? You have this portal triad at each of 6 corners of lobule. Real quick, hexagon, 6-sided. So each of the 6 corners of the lobule composed of following 3 structures: the hepatic arteriole, right? That's bringing blood; a portal venule, removing blood; and a small bile duct that carries bile. So your hepatic arterioles and portal venules both drain into large leaky capillaries called hepatic sinusoids. Right, these sinusoids have walls that are not continuous, allows large substances to enter and exit bloodstream. What could those large substances be? We'll be talking about those very soon. Blood flows slowly through the sinusoids as materials are exchanged between blood and hepatocytes, eventually draining into a central vein, which merges into the hepatic veins, and in turn feed into the inferior vena cava, that's blood coming from below the diaphragm back to the right atria. Bile flows to the liver lobule in the opposite direction, from hepatocytes into tiny bile ducts, eventually drain into bile duct. All right, so this is the basic lobule, the basic structure of the liver. Notice this central vein. This whole thing is the liver lobule. You have the bile duct, the green, the portal venule, the sort of the purple, and the hepatic arteriole, the red. All right. All right, what is the function? We see like the general structure. What is the function of the liver? And it is very diverse. Has releases both endocrine and exocrine secretions, converts harmful chemicals into non-toxic substances that the body can eliminate. Right, so it has endocrine, exocrine, and also can neutralize harmful substances. As I mentioned earlier, I have a family member who struggles with alcoholism, and this is one of the reasons why he's ended up with some liver problems, because your liver is responsible, sort of the organ that's responsible for when we put toxins into our body, such as alcohol, which is a toxin. We're not meant to overconsume. A glass of wine, I've heard, is okay, but and even that sometimes is questionable. But when we start to put too much in our body, and our liver struggles, and we'll see that here. All right, so the main digestive function is to produce bile, a liquid that contains water, electrolytes, and organic compounds. Bile serves two critical functions: digestion and absorption of lipids, fats. Remember, we talked a little bit about those when we talked about the bloodstream and the arteries and veins, and the other mechanism. The other purpose of bile is a mechanism by which liver excretes waste and other substances that kidneys cannot excrete. And we haven't gotten to the urinary system yet, but we will talk about the connection between the two. Bile salts, one of bile's main organic components, derived from cholesterol. They are amphiphilic. Remember amphiphilic when we talked about when in part one of this class, when you talked about the cell membrane. Amphiphilic means it has a polar side and a non-polar side. Allows them to interact with both lipids, which are non-polar, and a watery environment of the small intestines, which is polar. So these amphiphilic bile salts can react with the non-polar lipids and the polar water at the same time. Coastal lipids in the duodenum physically break them apart into smaller pieces, which is called emulsification. All right. And this is mechanical digestion, that's chemical digestion and absorption of lipids. So we breaking them apart basically makes them easier to digest. Bile also contains amounts of material the liver excretes, including cholesterol, waste products, and toxins. Most of these products are not reabsorbed, and they pass into the feces. Okay. Hepatocytes secrete bilirubin. And this is important, and hint, 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 I've seen a question on this on the final exam last year. The hepatocytes secrete bilirubin, which results, that's from the breakdown of hemoglobin by the spleen. Remember, red blood cells, erythrocytes go to the spleen when it's there, basically when it's time for them to be removed from the body. That's where they kind of go to die, so to speak. Hepatocytes secrete bilirubin from the which came from the breakdown of the hemoglobin by the spleen into the bile, right? So normal flora, remember that gut flora and large intestines, convert it to urobilirubin, then into stercobilin, responsible for the characteristic brown color of feces, right? I have that highlighted. Remember that, because you may see that again. That's where the brown color of feces it comes from. All right. So from bilirubin, which is from the breakdown of hemoglobin by your spleen, which is when blood cells go to your spleen and get broken up. The bilirubin gets secreted into your bile, which gets converted into urobilinogen, and then in the stercobilin, responsible for your characteristic brown color of your feces. Some urobilinogen is reabsorbed by large intestine and ends up back in the hepatic portal system. And then some remains in the blood and is excreted by your kidneys. And that gives some of that yellow color of urine, which you can easily get rid of if you drink enough water and dilute your urine. All right. So the breakdown of blood vessels, remember, blood cells only live about 100, a little bit over 100 days. Then we have to break them down. They help get that. We're constantly breaking down blood and building new blood cells. And that's what gives us the brown color of our feces and also the yellow color of our urine. Okay. Urobilinogen. All right. Some other functions of the liver: nutrient metabolism. All right. We have some processing of the nutrients we're going to talk about in the next section. Detoxification, detoxifies substances produced by body that are harmful. And excretion, directly excretes bilirubin in bile along with other substances. The gallbladder. Small sac sits on the posterior side of your liver. Just underneath you have your liver, just underneath you have your gallbladder. Receives most bile from the common hepatic duct, stores that bile, concentrates it by removing the water, and releases it when stimulated. Release again is stimulated by the CCK, all right, which also stimulates the pancreas. CCK triggers contraction of smooth muscle in the gallbladder, causing it to release bile into the cystic duct, joins with the common bile duct, joins with the pancreatic duct near the duodenum to form the hepatopancreatic ampulla. Ampulla is surrounded by a ring of smooth muscle, which has a sphincter, controls the emptying of bile and pancreatic fluids into the duodenum. Both of these enter in the duodenum. All right. Bile contains calcium salts and cholesterol. Both can precipitate and form hard lumps called gallstones. We don't want those. And so you see how your gallbladder, we have a picture. You can get gallstones in your gallbladder. That's from calcium. That's from calcium salts and cholesterol collecting in your gallbladder, right? Calcifying in your gallbladder. And the idea is that you want it to pass. You hope that they pass into your intestines and eventually out through your rectum. That would be the goal, but that doesn't always happen according to plan. All right. This is the basic structure. Looking if this is just a pretending that this was a real liver and you were sort of pulling it back, and you would see the gallbladder underneath, the cystic duct, the left and right hepatic duct coming from the liver, goes into a common bile duct, merges with the main pancreatic duct, and we get the hepatopancreatic ampulla right where it enters into the duodenum, first section of the small intestines. So the liver produces bile continually, but generally does not secrete it at a basal rate. Secretion occurs in response to gallbladder contraction, sphincter relaxation at the hepatopancreatic ampulla. And this is mostly accomplished by that same CCK. Stimulates that. And also the vagus nerve is involved. Other factors are that secretin, same secretin, and CCK that stimulate the pancreas, stimulate the liver and the gallbladder. But the most potent stimulus for bile production and release is bile itself. As bile salts re-enter the liver, bile secretion rises dramatically in positive feedback loop. Bile secretion continues into duodenum until duodenum empties, at which point CCK and secretin levels decline. Right, so the stimulus for the CCK and secretin levels, remember, is acidic substances, food coming into the partially digested proteins, also that acidic chyme coming into the duodenum. This stimulates the CCK and the secretin, and basically at the point to where all of that is cleared out, it moves due to motility, it moves through the intestines, moves out of the duodenum into the rest of the small intestines and eventually large intestines. When that happens, we stop the secretion of CCK and the secretin, and therefore stop the production of bile. Right? So duodenal cells secrete CCK and secretin into the blood. As we know, happens when food gets into this is what happens when food gets into the duodenum, because you have the acidic nature of it, the partially digested proteins, lipids. CCK triggers contraction of the gallbladder, which releases bile. Bile comes into the duodenum. Bile salts and secretin trigger the release of more bile. So bile salts in the intestines, that's absorbed by the bloodstream into the hepatic portovenual, that causes you to secrete more bile. Right? This is everything, the liver and the gallbladder, and basically all of your accessory organs of the digestive system put together. Right? We have the teeth, the tongue, salivary glands, pancreas, liver, and gallbladder. All assist in helping food be digested. For this chapter is a section on digestion and absorption. Remember, the goal of the digestive system is to be able to take nutrients into our body. So we eat food, we take it, it comes into our bodies, into our alimentary canals, technically, and then the nutrients from the food that we eat gets absorbed into our bodies and then helps our bodies to be able to function properly. Right? That is the goal. All right, so digestion breaks food down into smaller and smaller pieces. until only individual nutrient molecules remain. There's mechanical digestion. We talked about starting with chewing the food, and then we talked about the peristalsis and the churning and the segmentation. And then we also have chemical digestion. So not only do we break down the food mechanically, but we have chemicals, enzymes that react with the food to break apart the chemical bonds. Okay. So all nutrients go through the same mechanisms of mechanical digestion, mastication, churning, segmentation, and chemical digestion varies based on your nutrients. All right. So carbohydrates digest a little bit differently than proteins, which digest differently than lipids and nucleic acids, for instance. All right. Most digestive enzymes catalyze hydrolysis reactions. Hydro means we use a water molecule to break the bond between two molecules. Right? This explains why so much water is secreted with fluids like gastric and pancreatic juices. These water molecules are necessary to break down your food, right? You need water in order to break down food. When they talk about people losing weight, one of the things they say is to drink more water. Not only does drinking water give you a little bit more of a full sensation, so you don't eat as much because there's more, the water takes up some space in your stomach, but also water helps, your body needs water to be able to break down food. All right. And so to burn fat, you need to have some water in your body to break down the fat that you eat. To absorb it, you need to have water in your body. Right. And digestive enzymes speed up these reactions as well. All right, so this is an overview of the reaction. Most digestive enzymes catalyzed by these hydrolysis reactions. All right, so in this example, dipeptide, all right, polypeptides make up amino acids and make up proteins. And so we can use a water molecule, a hydrolysis reaction. We need a water molecule to help with an enzyme to help break apart this dipeptide into two individual peptides. All right. Once nutrients are digested into component molecules, then they enter the body. And we talked about this concept. The mucosa of the alimentary canal is an external membrane. So just because something is in the alimentary canal, it's considered an external membrane. Until your food is ingested, absorbed by the body, until it's absorbed by the body, it's not technically ingested. It's not technically in your body, right? Things, like I said, think of them, if you swallowed a really small marble, it would actually pass through your whole digestive system and it would come out in your bowel movement. It would not be absorbed into your body technically. Whereas the things that we do eat, carbohydrates, proteins, lipids, nucleic acids, are absorbed into our body. All right, so this is understanding absorption. Think of the enterocytes, which are the cells in your small intestines. All right, so to travel from the lumen of the alimentary canal to the blood, molecule must cross the barriers. Molecule being carbohydrate, protein, nucleic acid, lipid. These are examples. Sometimes small molecules can cross between enterocytes. So this whole thing is the enterocyte. Some small molecules can pass in between, like H2O, water. The apical side of the plasma membrane, but the general idea is this. Apical side of the plasma membrane, step one in figure, this side. So this is the inside of your small intestines. Some carbohydrate, for instance, first has to get through the apical side. Most substances require transport protein or channel to cross membrane. Channel or transport protein to get inside the cell of the enterocyte. if there is nothing to help it get into the enterocyte, then it will eventually remain in the intestines and eventually be excreted. But if it does pass into the channel, does pass through a channel or some transport protein helps it get inside, this is an active transport. Molecule travels through the cytosol of the enterocyte. Remember, it's a cell. These are the cells on the inner surface of the lumen inside the small intestines. Molecule exits the basal side, usually through another it can use another transport protein there. And then there's interstitial fluid between that and the blood capillary. Molecule passes through or between endothelial cells of the interstitial capillary and then enters the bloodstream. Right, so that's the path that nutrients from the food we've eaten, once they enter into the small intestines, has to take to get into our bloodstream, and then our bloodstream would take it to the necessary tissues in our body. Right, so now we're gonna break down each different type of substance: the carbohydrates, proteins, lipids, nucleic acids, because they're all a little bit different. Starting with carbohydrates, remember with carbohydrates, we start in the mouth. We eat something, you start mastication, chewing your food, you're breaking it down. But with carbohydrates specifically, you have salivary amylase enzyme secreted from your salivary glands in the mouth area, in the oral area, catalyze reactions that break long polysaccharide. Remember, polysaccharide is just a group, it's just a chain of monosaccharides, breaks them into shorter, what we call oligosaccharides. Right, so there's a little bit of chemical digestion, but there's a lot of mastication, a lot of mechanical digestion. And then the salivary amylase digestion continues in the stomach. But acid generally inactivates enzyme before it can complete tasks. The acid in your stomach will inactivate this enzyme before you really have complete breakdown of the carbohydrates. Chemical digestion of carbohydrates resumes in the small intestines, where the polysaccharides and oligosaccharides encounter the pancreatic amylase. So after they get into the duodenum, now we have more digestion of the carbohydrates. Oligosaccharide digestion is completed by reactions catalyzed by enzymes that are products of enterocytes on brush border or small intestines, includes lactase, maltase, and sucrase. Right? Lactase catalyzes digestions of the sugar lactose. We're going to talk about what it means to be lactose intolerant. It converts those into glucose and galactose. Maltase and sucrase catalyze reactions that break oligosaccharides into disaccharides and monosaccharides. So disaccharide, two different carbon chains, monosaccharide, 1, 5, or 6 carbon chain. So the end result of all this activity is monosaccharides, glucose, fructose, and galactose. So we've taken chains of chains of carbohydrates, carbohydrate chains. So these polysaccharides, broken them down into smaller, until eventually we get to glucose, fructose, and galactose, which your body can then absorb. So after carbohydrates have been digested means we've broken them down into glucose. Remember, glucose is a 6 carbon sugar, 1 single chain, the monosaccharide. After we've broken it down, they are ready to be absorbed at this point. So now they're small. When we eat carbohydrates, they're big they're big long chains, polysaccharides. Now we've broken it down into monosaccharides, much smaller chains. Glucose and galactose are transported across the enterocyte apical membrane by the same mechanism. Enterocyte, that's the cell on the inner surface of the lumen of the small intestines. All right, so concentrations of glucose and galactose are higher in enterocyte cytosol than they are in the lumen of the small intestine, so they're absorbed into the enterocytes. Absorption of the glucose and galactose requires these molecules to move against their concentration gradients. So it requires input of energy, active transport. Remember that. Energy in this case comes via secondary active transport mechanism known as the sodium glucose co-transporter. And it helps basically, it acts like you have the sodium potassium pump, which consumes ATP to drive sodium ions out of enterocyte into lumen small intestine, creates this concentration gradient, which helps this movement of glucose into and galactose into the enterocytes. Fructose, unable to bind the sodium glucose co-transporter, so it crosses the apical enterocyte, the inner surface of the enterocyte by a separate mechanism, binds to a channel that mediates its facilitated diffusion across the membrane. And this is a passive process, requires no energy. Means that you must have this concentration gradient present or fructose will not be absorbed. All right, so they all pass into so glucose, galactose pass into the enterocytes by an active mechanism. Fructose, facilitated diffusion, passes into the enterocyte based on concentrations. All three monosaccharides cross basal enterocyte membrane by the same facilitated diffusion mechanism. So once they pass through the basal side, is the side closest to the bloodstream, they use a facilitated diffusion mechanism, very similar to the one that helps fructose across the apical side. After the monosaccharides cross the basal membrane, they diffuse through the extracellular fluid into the capillaries. That's in your blood. And once they are in the blood, they're delivered to the liver via the hepatic portal vein for processing. Right, so this is just, and I think this is definitely easier to see when we look at it on a diagram. So we have polysaccharide, multiple chains, broken down into oligosaccharide, broken down to disaccharide, eventually into monosaccharide, smallest one. We have for glucose or galactose, this sodium pump, which helps it to, which is an active mechanism, requires energy, gets the glucose or galactose into the enterocyte. Fructose, on the other hand, has a facilitated diffusion mechanism. So based on differences in concentrations, passive mechanism, and then they both have the same facilitated diffusion mechanism on the basal side, close to the bloodstream, and they can eventually get out of the enterocyte into the bloodstream. Digestion, breaking down, and absorbing into the bloodstream, passing into the bloodstream. The term lactose intolerant simply means adults that lack the enzyme lactase and as a result cannot digest milk sugar, which is known as lactose. Right. So basically, what does that mean? You can't break down the carbohydrate in milk, the milk sugar, known as lactose, because you don't have the enzyme lactase. So you can't break down that. You can't break down that carbohydrate, that polysaccharide. Therefore, your body can't digest it. Very simply. Without lactase, disaccharides such as lactose cannot be absorbed into the enterocytes of the small intestines. Very simply put, that makes you lactose intolerant. Lactose in large intestines acts as an osmotic particle. If you remember osmosis, creates a gradient to draw water into the feces, leads to abdominal cramping and diarrhea. Diarrhea, remember, you have too much water in your feces. This is caused, being lactose intolerant can cause that to happen, more water to be drawn. Remember, osmosis, water moves in the direction of the highest concentration of solute. So this lactose in your intestines causes water to move into the intestines to try to dilute it. Therefore, you have more water in your feces and abdominal cramping and diarrhea. Lactose intolerant. All right, so let's move on to proteins. All right, so we talked about carbohydrates, now proteins, and these are all somewhat similar. All right, so chemical digestion of proteins does not begin until they reach the stomach, where they encounter pepsin. Right, remember pepsinogen, pepsin. So the chief cells of the gastric glands produce inactive pepsinogen, but when the pH drops to 2 because of the acidic stomach, it becomes pepsin. Right, so that's when we have digestion of your proteins. The pepsin helps digest your proteins. Activated pepsin catalyzes reaction that digests proteins into smaller polypeptides, oligopeptides, and some free amino acids. Pepsin reactions catalyze only 10 to 15% of proteins in ingested food. Majority of the protein digestion takes place in the small intestines with the help of your pancreatic enzymes and also your brush border enzymes. So 5 pancreatic enzymes, all released as inactive precursors. Right? We don't want, the reason why they're released as inactive precursors is to protect the pancreas from digesting itself. Remember, we're made up of proteins, so we don't want to digest our own. We don't want to digest our own bodies. All right. So first pancreatic enzyme to become activated is precursor trypsinogen, becomes trypsin when it encounters enzymes on the intestinal brush border. Catalyze reactions that convert other pancreatic enzymes to their active forms. These enzymes catalyze reactions that digest proteins and polypeptides. Okay, so you have some digestion of proteins in the stomach and then also in the duodenum, small intestines. Final enzymes that act on proteins are associated with enterocytes. So you have other enzymes from the enterocytes. There are multiple brush border enzymes that catalyze digestion of oligopeptides and the free amino acids. Remember, we're going to just break them down. Right, enzymes are limited in reactions. They're able to catalyze, so some small oligopeptides remain undigested. Right, so not everything gets digested, but these oligopeptides are taken up by enterocytes and cytoplasm. They encounter more enzymes and catalyze their breakdown into free amino acids. That's the smallest form that they can be broken down into. To be absorbed in the small intestines, proteins consumed for nutrition must be broken down into small oligopeptides and free amino acids. That's what we want to break them down to. Humans do have the ability to absorb small amounts of whole undigested proteins involved in immunity by process of endocytosis. You have specialized cells overlying your Peyer's patches in small intestines, endocytose. In small intestines, endocytose these proteins, deliver them to the lymphatic tissue in patches. Right, but what we want is these oligopeptides. In general, we want these oligopeptides and free amino acids, which cross the enterocyte apical membrane, that's on the inside, the lumen, primarily by this secondary active, that means requires energy, transport membrane proteins. Again, a sodium potassium pump. Within the enterocyte, these oligopeptides are broken down. So inside these cells on the surface of in the inner surface of the amino acid, we break down these oligopeptides into free amino acids. Free amino acids exit the basal side, the side closest to the blood, the basal side of the enterocyte by facilitated diffusion, very similar to your carbohydrates, after which they can enter the capillaries. And they're delivered to the liver for processing via the hepatic portal vein. And this is the diagram for absorption, digestion, and absorption of proteins, oligopeptides, chains of peptides, broken down into free amino acids, passed into the enterocytes. You have sodium potassium pump, creates this gradient, passes into gradient, drives secondary active transport, and then they eventually, these amino acid can pass through the basal side with facilitated diffusion. I mean, that means it's passive transport here at this point, active transport into the enterocyte, passive transport out of, and then eventually into the bloodstream and onto the liver. Liver's doing a lot of work here. Next, we have lipids. All right, so majority of your lipids, 90%, are triglycerides. Remember glycerol with 3 fatty acid chains, triglyceride, remember that structure? You learned that in part 1 of this course. The nonpolar, which is the fatty acid chains, nonpolar lipids tend to stick together. Remember, the fatty acid chains are chains of carbon with hydrogen. So the nonpolar lipids tend to stick together, forming large globules of fat, distributing evenly in a water-based solution. And they're broken down 2 ways. Mechanical digestion, we chew that fat up, start breaking it down, churning in the stomach, segmentation in small intestines. And then you also have bile salts, which in small intestines are amphiphilic molecules. We talked about those when we talked about the liver and the gallbladder. They're molecules with polar and nonpolar parts that can help with the emulsification of the fats. Here is bile salt structural formula. Polar end, that the polar end of the bile salt blends well with the water. Nonpolar end mixes well with the lipids, the nonpolar lipids. While bile salts mix with lipids, the nonpolar parts interact with lipids, while the polar parts interact with surrounding fluid, breaks up the lipids, and emulsify into smaller pieces by what we call emulsification. End result is multiple tiny lipid droplets coated with bile salts. Now the digestive enzymes can work on these surfaces. Right, so we have lipids broken apart by the stomach churning, broken down by gastric lipase. Lipids enter small intestines, are emulsified by bile salts. Pancreatic lipase catalyzes reactions that digest lipids into free fatty acids. Remember breaking it down and monoglycerides. So it's from triglyceride to monoglyceride. Bile salts remain associated with digested lipids to form what we call micelles. This is the process of lipid digestion. Started in the stomach, broken apart by stomach churning, broken down in reactions catalyzed by gastric lipase. Disaccharides, free fatty acids. They enter into the small intestines, emulsified by bile salts, lipids, bile salts, emulsified. Now they have surfaces that can be digested. Pancreatic lipase catalyzes reactions. Break them down into monoglycerides. And the bile salts remain associated with them, form the structure called the micelle. So that digests, so now we've digested them, now we have to absorb them. Due to their nonpolar nature, lipids are difficult. and it's difficult for them to pass into the cytosol of the enterocytes. All right, so the micelles escort lipids to enterocyte plasma membrane. Lipid pass through phospholipid bilayer in the cytosol, and then reassembled in the triglycerides and packaged in the chylomicrons. Right, these chylomicrons are released into the interstitial fluid by exocytosis. Notice not facilitated diffusion, and then enter lacteal. Lipids are not delivered directly to the liver via your hepatic portal vein after absorption. Right, it's a little bit different here, different from the proteins and the carbohydrates. This hepatic portal vein delivers leftover bile salts to the liver used to make new bile, but the lipids travel within the chylomicrons through lymphatic vessels. Remember, we talked about this when we talked about when we were in the immunity and lymphatic chapter. All right, so they don't pass, so they pass, the lipids travel through lymphatic vessels and eventually to thoracic duct, join blood with rest of lymph as thoracic duct meets junction of left internal jugular and left subclavian veins. As chylomicrons travel through blood capillaries, lipids are removed and enter cells where they can be used for a number of different purposes. Right, so they travel through lymphatic vessels and don't join with blood, and eventually to thoracic duct, join with blood and rest of and with rest of lymph as thoracic duct meets the internal jugular and subclavian veins. So they don't go directly into the bloodstream at the capillary level, right next to right next to the intestines, but travel through the lymphatic system and come in later. Right, so the micelle, enterocyte, bile salts break down, lipids enter enterocyte, form back into triglycerides in the enterocyte with the coating of the chylomicron. That moves out of the enterocyte by exocytosis, requires energy, and then enters into the lacteal, enters into eventually the lymphatic system. All right, so that's the absorption of lipids. Nucleic acids. Nucleic acids in food begin chemical digestion in the small intestines with help of your pancreatic enzymes called nucleases. Right? Catalyze reaction, break nucleic acids into individual nucleotides. Digestion occurs via brush border enzymes, remove phosphate group and sugar to leave phosphate ion, ribose, deoxyribose, and nitrogenous base. Remember, that's the structure of the nucleic acid. Absorbed via primary and secondary active transport mechanisms into your capillaries. Right, so just nucleases break down your nucleic acids into its different parts, and then it's absorbed into your body as necessary via primary and secondary active transport into your bloodstream. And this is all of your reaction necessary to digest and to absorb your different organic compounds, proteins, carbohydrates, lipids, and nucleic acids that we all eat in our food. Now, water, electrolytes, and vitamins. Now, obviously, it's important to drink water. Everybody is I'll be telling you that since you were young. Drink more water. It's very important. The alimentary canal, we talked about it when we talked about, especially in the large intestines, absorbs large quantities of water, electrolytes, and vitamins. Most occurs in the small intestines, but significant quantities occur in large intestines too. And so why do they say drink water? Over 9 liters of water enter the small intestines each day. 2 liters of water are ingested. That's what we drink, we're supposed to drink. Remaining 7 liters are secreted into alimentary canal by alimentary canal itself and accessory organs. Of this 9 liters, 8 liters are absorbed into enterocytes. Remaining water is absorbed into enterocytes of large intestines, leaving only about 0.1 liters of water to be excreted in your feces. Keep your feces a little bit moist. Water absorption occurs exclusively by osmosis. So it requires a concentration gradient, right? Remember, water moves from low concentration of solute to high concentration of solute. So the cytosol of the enterocytes and extracellular fluid in villi must be more concentrated than the fluid in the lumen, so the water will move into the enterocytes. Anything that causes excess solutes to remain in the lumen of intestines will hold onto water and prevent its reabsorption, right? Electrolytes are both taken in from diet and present in secretions from the digestive organs. Most of the electrolytes are absorbed in small intestines, although significant amounts are absorbed in large intestines. So your electrolytes absorbed in your small intestines, large intestines. Important mechanisms for absorption of sodium ions revolve around co-transport of monosaccharides and amino acids. So sodium is going to be a key electrolyte. Also helps in the absorption of anions. Okay. Vitamins, chemicals generally provided by diet that are involved in a host of metabolic reactions, help keep our body functioning properly. We have water-soluble vitamins, polar molecules, and fat-soluble vitamins, lipid-based, just like the sound, fat-soluble, lipid-based, mostly non-polar. Your water-soluble vitamins are absorbed in your small intestines by diffusing through enterocytes. Just the process of diffusion, that's passive, doesn't require energy. Except for B12, which must bind to an intrinsic factor, molecule produced by parietal cells of stomach to be absorbed in the ileum, right? Ileum, that's the last portion of your small intestines. Your fat-soluble vitamins are packaged into micelles with fats and other lipids and are absorbed with them. So absorbed, that's why you have to have some fat. Fat-soluble vitamins, A, D, E, and K. All right, so these are all how we get certain vitamins. So diffusion and certain vitamins we get with our fat. I'm not telling you to eat a lot of fat. We all probably eat it with more than enough fat. And then this diagram just kind of puts it all together.